All right, we're continuing with our four major organic molecules of life, and in this video, we are looking at proteins. Proteins are chains of amino acid monomers joined by, of course, are joined together by our friend, the dehydration synthesis reaction. They have a central carbon atom bonded to four other atoms or groups of atoms. So in the case of an amino acid will have a hydrogen atom. It will have an R group, which distinguishes one amino acid from another. It has an amino group. There's our NH2 and our carboxyl group, our COOH. Amino acids, again, are distinguished by their R group. That's kind of what makes them different. So they all have that central carbon. If you look at the 20 amino acids that are out there, every single one of them is going to have that central carbon atom. The R group will vary depending on which one you're looking at. So in glycine, we see that the R group is a single hydrogen. In alanine, it's a CH3. And then it goes on and becomes more and more complex with each one of these. So the R group is what makes an amino acid unique. One of the things to remember about amino acids, the human body and bodies of many animals can synthesize all but 8 of the amino acids, which means that our bodies can actually make the amino acids that we need. There are 8 of them, though, that our bodies cannot make ourselves, that our bodies can't synthesize them, so that our cells cannot make these amino acids. That means we have to consume them in food. And these will be high protein foods like red meats, or if you're vegetarian, maybe tofu, legumes, and things like that. So 12 amino acids the body synthesizes, 8 have to be consumed. Amino acids join together. When we're talking about a protein, remember that it starts with amino acids that are a whole bunch of them joined together by these dehydration synthesis reactions. So when they're joined together, they form this primary structure, which is just the polypeptide chain. A string of amino acids is called a polypeptide. So amino acids join together. So you have one amino acid joined to another is called a peptide. Multiple peptides joined together are polypeptides. So here we have a polypeptide chain. It then begins to take on a shape. So it's either going to coil or it's in a helix, like it's not true coil, it's a helix structure. Or it can fold kind of these zigzags, like if you were folding paper back and forth. And that's called the secondary structure. So the alpha helix or the beta sheet structures are the second phase of folding. So we're having these 4 phases that it takes for a protein to actually get to its final structure. And what a protein structure is what will determine its function, will determine what it can actually do. And that's really important because proteins do specific things, and an incorrectly folded protein cannot work properly. So we went from our polypeptide chain, folded into either beta sheets, alpha helix, or a combination. The tertiary structure is the overall fold of the protein. So now it's starting to move into 3 dimensions, and it takes on these, you see all these different secondary structures are actually kind of joined together and they're moving. It's difficult to see that this is 3-dimensional, but it is now a 3-dimensional structure. Finally, the quaternary structure is when different polypeptides have done all their folding, but now they are joined to other polypeptides to take on these very unique shapes that allow them to do very specific things. Examples would be enzymes that catalyze reactions in the body, and we'll learn a lot more about that moving down the road. But it's important to know now that a protein, the structure determines the function, and if the structure changes, the function is no longer able to occur. Well, look at me, not able to click. So when the shape of a protein is destroyed, it is called denaturation. So again, the shape determines the function. What are some of the ways that proteins can become denatured? Heat. Think about cooking an egg. It denatures the protein. So once you cook an egg, you cannot uncook an egg. It cannot get its shape back. Excess salt will denature proteins, cause it to lose its shape. It's one of the reasons that salting can be used to preserve food. The incorrect pH can denature proteins. So, for example, hydrochloric acid is an acid that's found in stomach acid. And while the stomach itself can tolerate that very low pH value, the acidity, other cells in our body cannot. So if they are exposed to the wrong pH, they will become denatured and break down. And you see that happening if you've ever seen someone who's like licking their lips in the wintertime because their lips are chapped and they accidentally chap the skin around the outside of their mouth. That's from the acid in the saliva that is denaturing proteins in the skin. And UV rays is another thing. So when you're in the sun, you see damage to skin done. UV rays can actually damage the proteins of the skin as well. And we'll learn that UV rays actually can denature DNA, which is even more serious. So all these can cause denaturation of proteins.